Okay, I'll real quickly go through some lower animal models, specifically flies, which is where a lot of our understanding of the innate immune system comes from. So Drosophila is, an incredibly tractable animal model, and it has an immune system. And a lot of that immune system is around the production of antimicrobial peptides, which are just literally peptides that you get secreted out of your cells, and they are damaging to various types of bacteria, just directly damaging. And flies make these in huge abundance, and they have a sensing and response system that will turn them on. And you'll see this sort of across all their systems here. The production of AMPs is sort of the number one thing that they can do. It's a fly. It doesn't really have like a ton of tools at its disposal, and it doesn't really need to live super long. And so it's not as sophisticated as any of the things we've been talking about. But they have things that look more or less like macrophages. And then they have cells that are involved in sort of clotting, which, if you watched the malaria lecture, platelets and mammalian systems can actually play an immunological role too. And so there's some analogies there. At the level of, but they have no adaptive immune system, no lymphocyte analog in any real obvious way. Very nice review here too about this. But as I mentioned, the toll-like receptor pathway really came out of discoveries from Drosophila. One of the guys that discovered toll was one of the Nobel Prize winners for their innate immunity when that happened. And toll is a Drosophila-like protein, the toll that looks like the toll-like receptors. Interestingly, instead of directly binding the pathogen molecular patterns like we do in mammalian cells, toll in flies binds this protein called spatzel that is actually an IL-17 analog. And so flies do have cytokine analogs. And interestingly, they have cytokine analogs that are sort of important for the big categories of things we've been talking about. So you have the toll pathway, which has this IL-17 analog, has proteins that bind pathogen-associated molecular patterns, but they're just higher up in the pathway. And then drives signaling very much like the signaling we see in mammalian cells in terms of NF-kappa B, MAP kinase activation, these degenerate pathways of cellular activation and transcription. And then it has the IMD pathway, which is the immune deficiency pathway. Everything in Drosophila is named sort of functionally and weird. And this is basically the TNF signaling pathway in mammalian systems. And again, turns on NF-kappa B type responses. And so, it can be a really useful, if you're looking at doing really precise perturbation-based hardcore modeling, Drosophila is a really great system to potentially consider doing it in because it's so tractable. And you'll have a lot of the same players, but they're not really put together in the same way. And the analogies are, not super well correlated. This just lists some of the cytokines that have been found. Like I said, there's an IL-17, there's a TNF. There's things like type 1 cytokines, so like the type 1 interferons. But interestingly, flies seem much more worried about bacteria than they do about viruses. And that could be an accident of evolution, but a lot of their focus is on getting rid of bacteria. And there's no adaptive immune system. And then I have one slide. I know you didn't ask about fish, but if you are looking for another really tractable small model, super, super tractable, zebrafish actually have a complete adaptive immune system. They have T cells, they do VDJ recombination, they have innate lymphocytes. This was only 2018 that it was really clear that the innate lymphocyte system, which is sort of these representative lymphocytes that make type 1, type 2, and IL-17 type responses, is really quite primitive and evolutionarily probably predates any other evolution in adaptive immunity. And so the great thing about zebrafish is if you want to sequence every T cell receptor in a zebrafish, that's real easy to do. You can grind up a zebrafish and get every T cell out of it in, like 5 minutes. Versus when we try to get all the T cells out of even a tractable animal like a mouse, that's a really huge production. It takes hours. It takes a lot of money. But the mouse is still closer to the human. But a lot of really great work with zebrafish, and particularly for years, there's been a lot of great work with TB and zebrafish. So that's all I had, and I'll hand it back. I'll answer any other questions, but then I'll hand it back to Andreas.